AI has become a tech bro buzzword. Did the Nobel Committee get taken in by the hype? This year, the Nobel Prize in Chemistry went to Demis Hassabis and John M. Jumper for protein structure prediction and to David Baker for computational protein design. These are two reciprocal fields. One tries to predict the amino acid sequence of a folded protein, whereas the other tries to predict how the protein will fold based on its amino acid sequence. These Nobel laureates all use computer models to solve these problems with machine learning techniques that we colloquially call AI. So let's talk about protein structures, sequences, and how this year's Nobel Prize winners help us to go back and forth between the two. Our DNA code for 4 nucleotides. Adenine, cytosine, guanine, and thymine are all A, C, G, and T. In the protein coding portions of our DNA, every 3 of those letters together make an amino acid, and we use an alphabet of 20 different amino acids to build our proteins. Plus another 2 we use but don't code for that are special cases. So, we have these linear coding sequences that make up our amino acid peptide chain, and yet, somehow, those chains fold up and link together and form 3D proteins. Proteins of a given sequence fold themselves to a consistent shape due to the order and chemical properties of their component amino acids. The polar amino acids tend to be on the outside of the protein structure where they interact with and bond to various components in the watery environment. Nonpolar amino acids tend to pack inward with other hydrophobic amino acids, creating tight formation that lends the protein structure. Polar amino acids can form hydrogen bonds, charged amino acids form ionic bonds, trade those electron bonds, and nonpolar cysteine can form very strong disulfide bonds. So as these different amino acids bond together at different points and as different polypeptide chains link up, the bonds between them fold the protein into its unique shape. All those chemical reactions between the amino acids are super complex, but they're consistent. A positively charged side chain is going to form a salt bridge with a nearby negatively charged side chain every time, like magnets. I know that's all a lot of chemistry talk, but the point is this. The linear sequence in our DNA, those A, C, G, and T's, should be approximately enough to give us the structure of the 3D protein. And it is enough. It's just really hard to predict all those complex chemical interactions from the sequence alone. Hopefully this also shows why protein structures get the Nobel Prize in chemistry when usually we think of proteins as biology. Really, this is biochemistry, and the interactions between amino acids are really operating on chemistry and physics principles here, so it's well within the scope of the chemistry Nobel. There are a lot of Nobel Prizes in chemistry and topics like these because bio and organic chemistry have a lot of interesting complex chemistry problems because, biology is complex. Anyway, back to it. So for years, we knew that sequence predicts protein structure. We just couldn't parse all that complexity accurately enough to take a reasonable guess at protein structures. And unfortunately, you can't just look at a protein through a normal light microscope to determine its shape because it is way too small for that. Since the 1950s, we've used X-ray crystallography to study protein structure. We've used it to understand the molecular structure of all sorts of biomolecules. It's the same technique Rosalind Franklin, put respect on her name, used to lead to discovery of key data that unlocked the structure of DNA. And honestly, if X-ray crystallography were really easy and we could just do it for all sorts of proteins, we'd probably just do that most of the time to figure out protein structure. But it isn't easy. X-ray crystallography is kind of wizardry. You got to get the protein to crystallize, and getting those little suckers to crystallize is one of those time-consuming, please let this work, please let this work. Dang, why the heck didn't it work? again kind of scientific protocols. I mean, it can take years to resolve the structure of a single protein. Other techniques like nuclear resonance and cryogenic electron microscopy cause less existential despair, but they also have limitations when it comes to large, complex proteins. So we have techniques to very accurately confirm a protein structure, but we're really sparing with them because it's such an absolute pain in the neck. And since we know that in theory, the DNA sequence alone should be enough to predict the protein's structure from all those chemical bonding relationships, if we can get our prediction accuracy high enough, we won't always have to confirm it for certain with X-ray crystallography, the science of pain. No, seriously, okay. There are professional crystallographers where this is their whole thing. They love their work. They're constantly figuring out ways to streamline their process. I'm not trying to razz them. My point is, in 2024, biologists can sequence DNA pretty much on a whim, but you have to really need to know the protein structure to do X-ray crystallography about it. It's really hard. Researchers built a bunch of tools to try to predict protein structures. For a long time, our computer modeling was so mediocre that humans could actually figure out protein folding better than our models. In fact, in 2008, one of this year's Nobel laureates, David Baker, helped found Foldit, a video game where humans folded proteins as a puzzle game. After all, we have a lovely, power-efficient brain computer with spatial reasoning and pattern matching capabilities. Over the years, the power of gamers was actually absolutely incredible. In 2016, Foldit players outperformed professional crystallographers and modeling algorithms when fitting a protein to its X-ray crystallography, and they repeated a similar feat in 2019. And hey, it feels good to see the intuitive power of gamers reign supreme, but this did still seem like the kind of job that machine learning should be good at. I mean, give it a bunch of sequences and their corresponding protein structures that we've already solved. Eventually, with the right model and enough training data, it should be able to predict the structure from the sequence. This is where Demis Hassabis and John Jumper come in. Demis Hassabis is a computer scientist and, a game dev, of all things. He was lead AI programmer for Black and White, a god game from Peter Molyneux's Lionhead Studio that I actually played when I was a kid. Demis Hassabis also founded a games studio and designed a couple strategy games, the Republic and Evil Genius series. But he discovered that game dev is a rough business. His studio closed down, and the dude just pivoted and got his PhD in computational neuroscience. Like, of course, you do. He then co-founded and became CEO of DeepMind, an AI startup, in 2010. Because Hassabis is an absolute gamer, DeepMind started working on AI that could play games, like Atari games, and that trajectory accelerated in 2014 when, yes, DeepMind was bought by Google. Under Hassabis, DeepMind put out AlphaGo, which outplayed the Go world champion. It put out AlphaZero, which is one of the highest performing chess engines. So it's a bit of a hard left turn when the DeepMind group suddenly get into the protein folding game. Now, the baby's first machine learning model of protein folding would probably go something like this. Grab the largest data set you can find of DNA sequences and known 3D structures that were solved by X-ray crystallography or similar. Train a model on that data set. Then, give it a new sequence and let it do some black box mystery magic, then spit out the folded protein structure as a result. It's the kind of model you learn when you're building an image classifier that tells you whether a photo contains a dog or a helicopter. And if that modeling approach worked, those algorithms wouldn't have been defeated by folded gamers. This is when they bring in John M. Jumper, the scientist who shared the Nobel Prize in Chemistry with Hassabis for protein structure prediction. Jumper has a master's in theoretical condensed matter physics and a doctorate in theoretical chemistry. You might be thinking that's the perfect background to model all these complex chemistry and physics of the amino acids that they interact to fold a protein. And guess what? The Nobel Committee apparently would agree. DeepMind's program, AlphaFold, uses an attention network, which relates the importance of different pieces of the sequence to each other and solves for them together. It's been described kind of like a jigsaw puzzle. You don't typically just lay out and then smoosh all the pieces at once into a solved puzzle. You might work the corners, find pieces that work together, relate them to each other. Kind of like the way the different amino acids relate to each other, forming bonds and fitting together chemically. So AlphaFold 1 comes out in 2018, and it's pretty impressive right away. It wins the big biannual protein folding competition, CASP, where protein structure researchers with a bunch of different models compete to see whose model could make the best predictions. At CASP, competitors are judged on proteins whose structure has been recently solved by X-ray crystallography or other techniques, but whose results haven't yet been published. So the right answer is known, just not known to the competitors. AlphaFold 1 wins in 2018. It's pretty cool. It's pretty impressive. But then AlphaFold 2 comes out a few years later. It's got a substantially different model under the hood, still a type of attention network, but it's not made of a bunch of separately trained modules like the first version, and its local physics-based adjustments work differently too. AlphaFold 2 just absolutely destroys at CASP the next time it meets. It predicts protein structures with over 90% accuracy. It is 5 times better than second place. So protein structure prediction is now AlphaFold and just everyone else. 2 years later than that, the AlphaFold team at DeepMind just drop a catalog of what would eventually be 200 million protein structure predictions. These obviously aren't confirmed protein structures, but they're AlphaFold's best guesses, and apparently AlphaFold's guesses are really good. Protein structure researchers are absolutely floored. They're saying things like protein structures are a solved problem. It feels like the end of history for this field. It is the single greatest leap the field's ever seen. AlphaFold 3 came out earlier this year, and the AlphaFold server is available to researchers. And its predictions are now comparable to experimental methods. It's not to say AlphaFold predictions are the same as experimental methods. X-ray crystallography is looking at the actual protein for its actual structure. It's not the concept of a structure, like a prediction, but experimental error also exists, however small. And as AlphaFold's prediction error rate approaches the experimental error rate of those techniques, prediction becomes good enough and, oh my god, so much easier. Some researchers are going to need to know the experimentally confirmed structure of a protein by X-ray crystallography, cryo-EM, NMR, all those kind of techniques, but that's only for some specific use cases. For other biology research, AlphaFold's good enough is enough for their experiments, and it's much more accessible to the average biologist. And protein structure prediction help feeds into that reciprocal we talked about at the beginning of the story, which accounts for David Baker's part of this year's Nobel Prize in Chemistry. Protein design. Because form is function for a protein, if you want it to do a certain thing, it's going to have a certain kind of structure. Proteins typically interact with other proteins in a sort of lock and key kind of way. So with protein design, if you want protein A to activate protein B, you gotta make protein A into a shape that can lock in with protein B. Protein design can also make more effective versions of existing proteins from nature. That is, making a key that's a better fit for the lock. After all, evolutionary pressure works on a strictly... good enough, send it. Standard. If a protein coding gene doesn't substantially affect your ability to pass on your own genes, there isn't going to be positive selection toward altering its function any further. That doesn't mean that there aren't benefits to these kind of protein redesigns, as they're called. But David Baker's group did more than redesign existing proteins, which, to be fair, we've been doing for quite a while. No, Baker's group were the first to design a novel protein, completely from scratch, that was not based on a pre-existing protein. They've gone on to build several thousand new proteins, catalyzing chemical reactions, neutralizing viruses. They even created the first on computationally designed protein as medicine, a protein vaccine against COVID. Some of his work has been used with machine learning models, others used computational but non-machine learning techniques. The techniques from these respective prize winners feed back and forth into one another, as protein structure prediction sure as heck empowers protein design. And to be clear, predicting protein structures and designing our own proteins absolutely changes the game in this corner of biochemistry, biology, bioengineering. It's like unlocking a whole skill tree in the game. Designed proteins unlock a range of experiments and medicines that aren't possible until now, and it's a toolbox that can synergize with all sorts of other techniques that we have in these fields. This year, the Nobel Prize in Physics also went to work in machine learning and artificial neural networks, so some scientists have expressed concern that it was just a big AI hype year for the Nobel Committee. Whether or not the committee hyped too close to the sun, the truth is, these prizes in chemistry are eminently deserved. But these Nobel laureates aren't the only ones catching strays from AI backlash or cashing in on the hype. Because I cannot stress this enough, ChatGPT is not built to do anything with proteins. It's a chatbot. It's trained on data from the public internet. I'm sure it could very confidently hallucinate about protein structure. But you cannot use it for the things that won these Nobel laureates their prize. That said, you can totally string together models like little string lights and put a chatbot on the end to present the results in friendly human words. Even ChatGPT does not actually use ChatGPT in any way. This is a program that uses an open source model to try to make sense of a human prompt, or it accepts an amino acid sequence. Then these things will hand off any real protein work to a dedicated module that knows what it's doing. It's a protein research clippy. And I get it. If I were working in bioinformatics and biochemistry and I were using machine learning, I would absolutely be trying to ride the hype wave. These scientists have grad students and postdocs and lab staff to look out for. They gotta get their funding, and ChatGPT for proteins, like, Uber for dogs to a venture capitalist, get some attention. But while slinging around the name of ChatGPT makes for good marketing for the university newsletter, this year's Nobel Prize shows that the stuff we call AI is so much more than the popular wave of generative AI web scrapers of the past couple years. Machine learning has been huge in biochemistry and bioscience for years, quietly transforming protein structure prediction from a thing we play video games about and can only confirm through laborious imaging, to a thing researchers can use software for to get accurate enough guesses that it's actually practical, and maybe sometimes we can avoid the X-ray crystallography, please. That would be so nice. The Nobel Committee was not taken in by hype in chemistry. This year's laureates in chemistry have genuinely transformed how we do science in these fields in ways that have nothing to do with generative models trained on copyrighted data from the Common Crawler dataset or similar web crawl approaches that power large-scale generative text and text-to-image models. This year's Nobel Prize in Chemistry should be a shining example of what we want from advances in this technology, irrespective of the clickbait AI buzzwording around it. Rational protein design is brilliant. Protein structure prediction is fantastic. Take away the buzzword and you have scientific findings that genuinely benefit humankind. That's pretty prizeworthy to me. The gamers still win.